We had some major medical breakthroughs over the past several centuries. For example, the discovery of vaccinations led to the elimination of smallpox from the surface of the earth. Contracting HIV used to be a death sentence in the past, but HIV medications allow HIV-positive people to live as long as a person without HIV. It seems like scientists have been able to come up with ways to prevent or treat diseases that once had no treatment option. But have you ever thought if a disease that once had a cure cannot be cured anymore in the future? Well, for some diseases, we may be heading in that direction. Another example of a medical breakthrough was the accidental discovery of antibiotics by Sir Alexander Fleming in 1928. One day, Dr. Fleming noticed something unusual on one of his petri plates. A mold was growing on it. He noticed that there was a clear zone immediately around it, as if the mold had produced something that inhibited bacteria to grow. Later, it was identified that this penicillium mold was secreting a substance called penicillin. Since the discovery of penicillin, antibiotics have saved millions of lives. Penicillin played a critical role during World War II. In previous wars, soldiers were most likely to die from infections than from the wounds. In fact, the wide-scale production and use of antibiotics increased human lifespan by 8 years between 1944 and 1972. That's remarkable. However, soon after the discovery of penicillin by 1942, doctors have noticed that penicillin was not working as it used to. This phenomenon is called antibiotic resistance. Antibiotic resistance is the bacteria's ability to resist the effects of an antibiotic to which they were once sensitive. Notice that bacteria, and not humans or animals, become resistant to antibiotics. Antibiotic resistance is one of the biggest public health challenges that we are facing now. According to the CDC, Centers of Disease and Control, at least 2.8 million people acquire an antibiotic-resistant infection, and more than 35,000 people die from it each year in the United States. If you have heard of MRSA or C. diff, those are examples of antibiotic-resistant bacteria. Some people may feel that getting an antibiotic-resistant infection is rather uncommon. A survey done by the WHO, World Health Organization, revealed some misunderstandings on antibiotic resistance. 2/3 of people believe that individuals are not at risk of getting a drug-resistant infection if they personally take their antibiotics as prescribed. In addition, nearly half of people think that antibiotic resistance is only a problem for people who take antibiotics regularly. The truth is, anyone of any age in any country can get an antibiotic-resistant infection. The CDC has identified 5 antibiotic-resistant microorganisms that are considered urgent threat to our public health. Actually, one of them is very commonly seen within our community. That is, Neisseria gonorrhoeae, the bacterium that causes gonorrhea. Gonorrhea is the second most common bacterial STD, sexually transmitted disease, in North America. CDC estimates that in the United States each year, there are 1.14 million cases of gonorrhea infections, and within that, 550,000 are drug-resistant. In case you were wondering, the name comes from the Greek gonos, meaning seed, and rho, meaning flow. Early physicians thought that the purulent discharge was semen. The name Neisseria comes from Albert Neisser, who discovered the bacterium in 1879. The bacterium has a diplococcal shape, meaning 2 spheres are attached to one another. You can see the bacteria as small dots if you look under the microscope. Why is gonorrhea an urgent threat to our community? The major reason is because this bacterium quickly develops resistance to antibiotics, and it is indeed resistant to most of them. Looking at the graph, you can see that the bacterium is increasingly becoming resistant to antibiotics like tetracycline, ciprofloxacin, and penicillin. In fact, Ceftriaxone is the last recommended antibiotic to treat gonorrhea. Furthermore, gonorrhea spreads easily because some men and most women do not have symptoms, and they may not know that they are infected. If the bacterium becomes resistant to antibiotics like ceftriaxone, gonorrhea may someday become a disease that cannot be cured anymore. Then, how do bacteria get this superpower that allowed them to be resistant to antibiotics? First, let's take a look at how a bacterium obtains antibiotic resistance in the first place. Bacteria, and as humans and other organisms, store their genetic information in DNA. Using the information, we make structures that are necessary to our body. Antibiotic resistance occurs through random, spontaneous errors in this process, called mutations. If there is an error in the genetic information, the body may make altered products at the end. For most cases, effects of mutations on the organism is neutral, but for some cases, these mutations can be beneficial for the organism, like antibiotic resistance. These beneficial mutations in DNA ultimately lead natural selection, the process whereby organisms better adapt to their environment, tend to survive and produce more offspring. This theory was first developed by Charles Darwin and is now believed to be the main process that brings about evolution. So let's see how natural selection is applied to antibiotic resistance. Let's say you get a bacterial infection. Some of those bad bacteria may undergo mutations, causing to have that antibiotic resistance superpower. You go see a doctor, they prescribe you antibiotics, and you take them. The antibiotics kill the targeted bad bacteria, but they may also kill your good bacteria, like the ones in your gut. This is why you may have diarrhea when you are taking antibiotics. Notice that the bad bacteria with the antibiotic resistance were able to survive. Those bacteria will continue to reproduce, causing you to still have that infection. Furthermore, a bacterium can obtain antibiotic resistance from another bacterium, mostly in two ways. A bacterium can go through the process called transformation, where it takes up antibiotic resistance DNA from another bacterium that has lysed or cut open. Or sometimes, the antibiotic resistance DNA can be included in a structure called a plasmid. Plasmids are small circular DNA molecules, and a bacterium with the antibiotic resistance plasmid can give copies of the plasmids to other bacteria. This process is called conjugation. Now that we know how bacteria can obtain antibiotic resistance, we can now learn how antibiotic resistance works. I'm going to compare the effects of two antibiotics on the gonorrhea bacterium. I'm going to use ceftriaxone to explain how antibiotics kill bacteria, and use penicillin as an example of how bacteria can be resistant to antibiotics. Looking at the two molecular structures, do you see any similarities? These antibiotics are in a group called beta-lactam antibiotics because they have this beta-lactam ring in their structures. There are two interesting things about the beta-lactam ring. The ring is made of two amino acids, valine and cysteine, and we can see the resemblance in the structure. In addition, when atoms are attached to four other atoms, they usually like to spread out in a way that it has a bond angle of 109.5 degrees. But in the beta-lactam ring, the bond angle is 90 degrees, producing a strain within the ring. This information may be too technical, but what's most important to understand is that this angle strain makes the antibiotic more reactive, allowing it to do its job. If you zoom into the cell wall of the gonorrhea bacterium, you will see two membranes. In between them, you will find a layer called peptidoglycan. Peptido meaning peptide or chain of amino acids, and glycan meaning sugar. So peptidoglycan is made of sugar molecules with peptide chains coming off of them. The peptidoglycan layers are linked with one another through the cross bridges of the amino acid chains, and this is done by the enzyme called penicillin binding protein, or transpeptidase. These cross bridges make the cell wall stronger. Let's see how ceftriaxone works to kill bacteria. Antibiotic molecules enter the bacterial cell through structures called porins that are on the outer membrane. Remember that the ring strain on the beta-lactam ring makes the molecule more reactive? The antibiotic molecules permanently bind to the penicillin binding proteins, preventing them to make cross bridges of the peptide chains. The cell wall becomes weaker, causing the cell to burst and kill the bacterium. Penicillin was used to treat gonorrhea infections starting in the 1940s, but the era of using penicillin to treat gonorrhea ended in the mid-70s. There are 4 features why penicillin is not effective against gonorrhea anymore. Those are the use of beta-lactamase, changing penicillin binding protein shape, decreased permeability through porins, and the overexpression of efflux pumps. Neisseria gonorrhoeae has acquired a plasmid from another bacterium that encodes the enzyme called beta-lactamase. This enzyme hydrolyzes the penicillin's beta-lactam ring, meaning it uses water to open the ring. This ring opening makes penicillin inactive. This beta-lactamase cannot make ceftriaxone inactive, but some bacteria produce an enzyme called extended-spectrum beta-lactamase, which can hydrolyze ceftriaxone's beta-lactam ring. If the gonorrhea bacterium obtains a plasmid that encodes extended-spectrum beta-lactamase, ceftriaxone may not be used to treat gonorrhea anymore. We have learned that mutations in the DNA can produce products with different shapes. Mutations in the DNA where it codes the penicillin binding proteins cause these structures to have a different shape, and this decreases the rate of these enzymes to bind with penicillin. The penicillin's work cannot catch up with the rate of the bacteria making the cross bridges. If there is a mutation in the DNA where it codes for porins, the structure of the porins can change so that they become less permeable, preventing penicillin molecules to enter the cell. Lastly, mutations not only can cause to make different shaped products, but also can make too much of the product. Mutations in the DNA can overexpress structures called efflux pumps. Bacteria use these pumps to eliminate toxic substances like penicillin. Efflux pumps export the penicillin out of the cell, so even if penicillin molecules enter the cell, they will be simply transported out. According to the WHO survey, 57% of people feel that there is not much that they can do to stop the spread of antibiotic resistance. Well, I'm here to tell you that everyone can help to prevent the emergence of antibiotic-resistant bacteria. But first, let's evaluate how you're using antibiotics. Do you ever stop taking antibiotics when you start to feel better? Do you ever take antibiotics for a common cold or flu? Do you take leftover antibiotics from a previous infection? Do you take antibiotics that were prescribed to someone else? If you are, please stop now, because you're using antibiotics inappropriately, and these actions promote antibiotic resistance. A person may inappropriately use antibiotics by overusing or misusing them. Antibiotics can be overused when a person takes them when they're not needed. Antibiotics only work on bacteria, so taking it for other types of infections, such as for viral infections, will not work. Antibiotics can also be misused when they are taken with the wrong antibiotic, duration, or dose. Some people stop taking antibiotics when they start feeling better. When people do not take the antibiotics for the whole prescribed duration, the most resistant bacteria will survive. Also, when some people feel that they need antibiotics, they sometimes take leftover antibiotics from a previous infection or take someone else's antibiotics. The antibiotics they are taking may not be the right kind to treat the infection. So, what can you do? Ultimately, you can do your best to use antibiotics correctly. If you were prescribed antibiotics, take as directed and finish the whole course. Ask your doctor or pharmacist first if you feel that you need antibiotics, and don't self-diagnose and self-prescribe antibiotics. Remember, antibiotics only treat bacterial infections, and they do not treat viral infections. Everyone in our community has the responsibility to use antibiotics correctly, so that we can slow down the process of antibiotic resistance. I would like to end with a quote from the CDC. Everyone has a role to play in improving antibiotic use. Appropriate antibiotic use helps fight antibiotic resistance and ensures these life-saving drugs will be available for future generations. I would like to thank Dr. Stephen Farmer at SSU, who gave me guidance and support throughout my research project, and Dr. Katie Jamshidi at SRJC, who made me realize how fascinated I am in microbiology. Lastly, I would like to thank my parents and friends who gave me feedback on this video. This was Aiko Tompkins, and thank you for listening.